Hi everyone, how are you feeling? I hope you're feeling good and ready to finish off our last section of microbiology. Yay! Clearly I am so excited. Okay, so here is our mind map of what we have covered thus far. Okay, so just have a look at those, and this is last time in our last recording we got up to the chain of infection, and then before that, so the last recording was in green, the one before that is in orange, and then the one before that is in black. So we've covered a lot, hey? Now this is a way that I kind of put it together. Please feel free to change it and make it work for you. Okie dokie. So let's talk about infection, okay? I do have some notes here because I want to make sure that I tell you the correct information, okay? All right. Infections. This is very, very important, clearly, when you're going to be a health professional, right? So it's very important that you understand the basics of infection. Okay, now, infection, that term, is when you have an infectious agent, okay, that goes into the human body, but not only does it go into, it actually grows and develops and sets up a house there, a colony, okay? That is what an infection is. An infectious agent going into your body and growing there. And an infection can or cannot cause a disease, because we, as you know, we have lots of microorganisms that live in us, okay? So an infection is an entrance and development of an infectious agent in a human body, whether or not it develops into a disease, okay? Because you can be infected and that cannot develop into a disease, okay? So just be mindful of that. What is a sign? What is a symptom? A sign is your measure, okay? Objective. Symptom is what is subjective, is what the patient is telling you, okay? Just be aware of that. All right, so let's have a look at the stages of infection, okay? Very, very, very important. Now, what I want you guys to look at in particular is, look at this. This is the number of disease-causing microorganisms, okay, or particles. So can you see here, look, that's the red one. Look how many, and they go up because they divide, they make a little community. And then as we start to kill them off, they go down in number, okay? And look at that. Look at this blue graph. It mirrors the red one, because the more pathogens you have in you, the worse you're going to feel. It's a direct correlation. Simple as that. So let's have a look and see what each of these stages represents. The first stage here is called the incubation stage, right? This stage is just you are being exposed to the infectious agent, okay? But you don't have any symptoms yet, all right? So you have been exposed to the microorganism. It's in you, and then it's going to start making copies of itself, all right? So that is the incubation stage. The prodromal stage, all right, in this particular stage, the microorganism keeps replicating, all right? It's starting to, the community of microorganisms is growing bigger. At this point, your immune system catches on, and then so you start getting mild, non-specific symptoms. It's like your immune system goes, hey, wait a minute, there's something going on here. We don't know what it is yet, so you're starting to feel that, okay? The third part of the disease or the infection is called the period of illness. Okay, now you are starting to show symptoms of that particular infectious disease. Okay, now clearly, look, this is when you have the highest number of microorganisms or pathogens in you. You're not going to feel good. Okay. The next stage is that your immune system finally catches on to what's happening and starts to attack whatever the microorganism was. Okay? So the period of decline means that your immune system is fighting to get the number down of this microorganism in you. Okay, can you see that it takes a little bit for your immune system to catch on to see what's going on? Okay. The final stage is called convalescence, and at this point your immune system is kind of nearly killed all the microorganisms off, and that means that your symptoms are pretty much going to go away. Okay, and essentially that is what an infection looks like. All types of infections have the same stages. It's just the time that it takes for each stage is just different for each different infection. So for example, this is just an example that the incubation period is different for different infectious diseases. Okay? So just be aware of that this is the general kind of pattern, but for each infectious disease, their stages have different times, lengths of time, but it all follows this same format. All right, I'm happy with that. Make sure I told you the right information. All right, so what does the word pathogenicity mean? Pathogenicity means whether or not a microorganism can cause disease, okay? Now there are two options there. Why is my camera trying? There's two options there. Your microorganism can cause disease or it can't in humans, okay? So there's two options. Virulence only applies to those microorganisms that can cause disease. So you're like, okay, this microorganism is pathogenic. Okay, let's see how bad it is. So virulence describes how bad or the severity of the disease that particular pathogen can cause. Okay? Now, there are things called virulence factors, which basically means that if you are a microorganism and you have these, it's going to make the disease that you cause worse. Okay? So endotoxins, when you have a bacteria living inside of your body, if you kill it, sometimes the cell wall comes apart and that makes you sick. That is an endotoxin. An exotoxin is made by the bacteria while it's still alive, and that also makes you sick. Okay? So just be aware of the fact this is extra. So if I'm a bacteria and I make exotoxins, I'm going to be more capable of causing disease than one that doesn't. Okay? These are all little tricks that these microorganisms have up their sleeve to make us feel worse. Another thing, you can have a capsule, which is like an extra coat of armor on the top of your cell wall, which makes you harder to kill. Okay? You can have spikes, which make you stick onto surfaces much, much easier, okay? You know, so there are little tips and tricks that bacteria have that make them A, harder to kill, B, release stuff that's going to make us sicker. All right, so now that we've learnt about microbes and how they can infect us and actually make us not feel too great, what we can do is think about how we can kill microorganisms that are pathogenic, or how we can at least, you know, kind of get a hold of or control how much they grow, okay? Now, there are different things that can affect the growth of microbes, and they are listed there. So, for example, let me just go through here. So, if you are a microbe and you don't have enough to eat, then you're not going to grow as much. Make sense? If if I'm a microbe and the temperature is too hot that it burns me, clearly I can't grow. Okay, so all of these things are just things that you need to be mindful of when you're looking at a microbe. A microbe is just like a person. It needs the right nutrients, it needs the right temperature, it needs the right oxygen requirements. It's fussy. It has its things that it needs for it to grow. And if we change something like that, then we're going to change the growth rate. Remember that when we are talking about microbial growth, it is them making copies of themselves, not necessarily them getting bigger. Okay? All right. So, you know, like we know that microorganisms just make lots and lots of copies of themselves, and going from one copy to 2 copies is called something called generation time, and it can take, you know, a very short amount of time to get lots and lots and lots of copies. Okay, so yeah. All right, very, very, very important. Okay, because you as health professionals need to know and understand these terms. There's only 3 really that are hardcore important. When you sterilize something, you are killing, okay, you are eliminating all forms of microbial life, which means both vegetative and spores. The vegetative ones are bacteria that are still growing, that are still multiplying. And the spores are like the endospores that they make when they know they're under threat and they want to put their little DNA into a little capsule and, you know, kind of ride out the storm until it's all over, and then they can become active again, okay? So you're killing the cells that are active and their dormant little kitties or little supermans in the pods, okay? That's what you're killing off when you're doing sterilisation. You're getting rid of all of it. Disinfection eliminates everything but the spores. And you're doing it to an inanimate object, which is different to antiseptics. Okay? Let's have a look at a picture. Look at this. Sterilisation kills everything. The cells that are alive and active and making spores, and the spores that are hanging around there for just in case the other guys get killed off. Okay? Disinfection, okay? We don't kill off the spores in this instance. Okay. Now there's different ways, different methods that we can use to sterilise and disinfect. We talked about autoclaves already in class. You can put alcohols, okay? There's different ways that we can actually, kind of disinfect and sterilise, but you need to know the difference, okay? Now, we already know what disinfection meant. We're going to eliminate the cells that are alive, and we can't get rid of the spores from inanimate objects. So antiseptic is just doing disinfection but on people. on human skin. Okay, that's basically the difference. Yeah. Okay, one more set of terminology. If something is microbacidial, that means it's going to kill microorganisms. If it's called microbistatic, it's not necessarily going to kill them, it's going to stop them making copies of themselves. That's pretty much it, and we have finished microbiology. And don't forget to add what we just talked about onto our mind map here and play around with it and make your own. And yeah, that is it.